Hello everyone, welcome you all in the EPG Pathshala program in the subject botany. Today I will talk on ethylene. Ethylene is a colorless, flammable and gaseous organic compound with a sweet and musky odor. Though ethylene has been used to stimulate the ripening since the ancient Egyptians, the effect of ethylene on the plant growth was noted for the first time in the year 1864 when a leakage of gas from the street lights showed stunning growth, twisting plants and abnormal thickening in the stem. Ethylene is also a naturally occurring plant hormone and influences many plant processes ranging from germination to senescence. In this module, we will study how ethylene is synthesized and metabolized. We will also see the physiological effects that these hormones has on plants, signaling and regulation and its crosstalk with other hormones. Chemically, ethylene is an unsaturated hydrocarbon with carbon-carbon double bond. In higher plants, ethylene is synthesized in all the plant parts, namely roots, tubers, stems, leaves, buds, flowers and fruits. Now, we will see how ethylene is synthesized in plants. Ethylene is biosynthesized from methionine via S-adenosyl L-methionine abbreviated as S-adomate and 1-aminocyclopropane 1-carboxylic acid, that is ACC. The formation of methionine to adomate is catalyzed by the enzyme S-adenosyl L-methionine synthetase, also known as SAM synthetase. For the synthesis of one molecule of S-adomate, one molecule of ATP is required. Further, S-adomate is converted to ACC by the enzyme ACC synthase. This is the rate limiting step in ethylene biosynthesis. The final step of ethylene biosynthesis is catalyzed by the enzyme ACC oxidase, which converts ACC to ethylene and generates carbon dioxide and cyanide. To prevent toxicity of accumulated cyanide during high rates of ethylene synthesis, plants detoxified it to cyanoalanine by the enzyme cyanoalanine synthase. Earlier, it was believed that ethylene is inert. However, ethylene is rapidly metabolized to ethylene oxide by the enzyme ethylene oxide, which is a monoxygenase. This compound can be readily converted to ethylene glycol either enzymatically or non-enzymatically. Ethylene glycol is the stable end product of ethylene metabolism. After knowing the biosynthesis of ethylene, we will see the physiological effects of ethylene. Ethylene influences plant processes such as seed germination, growth, diageotropism, senescence, flowering, abscission, fruit ripening, abiotic and biotic stress responses. First, I will talk about effect of ethylene on seed dormancy and germination. We know that seed dormancy prevents germination during the unfavorable period. This allows the seed to survive after being dispersed from the mother plants. Ethylene is produced during germination and the production of ethylene is stimulated by the factors that break dormancy. Ethylene can stimulate germination even at very low concentration. However, in few species, ethylene has no effect or adverse effect on germination. ACC, the immediate precursor of ethylene, can also stimulate the germination. However, the effect of ethylene on germination is more pronounced than ACC. Together, these facts suggested that seed's inability to convert ACC to ethylene is responsible for reduced ethylene production that inhibits seed germination. Ethylene also influences plant growth. Ethylene inhibits growth by impairing cell division, DNA synthesis and cell expansion in the meristems of roots, shoots and axillary buds. Application of ethylene to etiolated seedlings causes 3 distinct morphological changes, which is called as triple response in the seedling. Number 1, inhibition of stem elongation, 2, radial swelling of the stem, and 3, absence of normal geotropic response, that is diageotropism. Ethylene also reduces the activity of DNA synthase, therefore, DNA synthesis is inhibited. Interestingly, ethylene was the first plant hormone identified as a senescence accelerator. Now, we will see how ethylene activates the transcription of different genes in plants. First, in the membrane of endoplasmic reticulum, ethylene is perceived by its receptor ethylene response 1 or ETR1. Upon ethylene binding, ETR1 is unable to activate a serine or threonine protein kinase constitutive response 1 or CTR1. Therefore, CTR1 stops phosphorylation of the protein ethylene insensitive 2 or IN2. Non-phosphorylated cytosolic C terminal of IN2 is eventually cleaved by the protease. Further, C terminus of the cleaved IN2 is moved into the nucleus where it activates the transcription of different genes. Ethylene plays a significant role in flower initiation. In some species, ethylene inhibits flower induction and enhances flower bud abortion. On the contrary, in some species, ethylene promotes flower bud induction. Ethylene production inhibits CTR1 and enhances IN3 levels via the SCF ubiquitin pathway and activates the ethylene signaling. Activation of ethylene signaling reduces the accumulation of bioactive GA and increases the accumulation of DELA protein. Enhanced DELA accumulation inhibits the suppression of floral meristem identity genes called as LEAFY or LFY and suppressor of overexpression constants 1, that is SOC1, which in turn delay flowering. Ethylene stimulates flowering in pineapples, other bromeliads, mangoes and in some geophytes. Ethylene also influences ripening of fruits. Fruit ripening is a genetically programmed event. Fruit softening, one of the ripening processes, is very sensitive to ethylene. A developmentally regulated increase in the expression of both ACO and ACS initiate a rise in the ethylene production. However, changes in fruit color can be ethylene dependent or independent. Several ethylene responsive fruit ripening genes are identified, namely E4 and E8. Next important function of ethylene is acceleration of abscission. Abscission is the process by which plants shed its different parts. Combined activities of several cell wall degrading enzymes are responsible for the changes required for abscission. Cellulases and pectinases are the major ones. Ethylene accelerates abscission of leaves, flowers and fruits by regulating the expression of genes of these enzymes. In addition to cellulases and pectinases, ethylene also enhances the growth of the cells in the abscission zone, but not in the neighboring cells. This uneven growth generates the mechanical force. Mechanical force in combination with the weakened cell wall allows the shedding of the plant part. In response to variety of stresses like low temperature, osmotic stress, chemical stress, mechanical stress, gravitational stress, wounding, radiation, ozone, heavy metals, herbivory and pathogen attack, plant stimulates ethylene production. Ethylene elicits plants' defense responses by enhancing the emission of volatile organic compounds, accumulation of phenolics and activity of proteinase inhibitors. Ethylene shows a dramatic effect on the expression of plant defense response genes, namely 1L-phenylalanine ammonia lyase and 4-coumarate CoA ligase enzymes of the phenylpropanoid pathway. Second is chalcone synthase, an enzyme of the flavonoid glycoside pathway, and third is the hydroxyproline rich glycoprotein, a major protein component of the plant cell walls. We have already seen that ethylene production is regulated by differential transcription of ACS gene. In most species, ACS is regulated by multigene families. The expression of different ACS genes are specific to the developmental stages. The use of ethylene in sensitive mutant revealed that among all the ACS genes, only LEACS2 expression is ethylene dependent. In climacteric plants, two systems of ethylene production have been proposed. System 1, according to this system, ethylene inhibits its own biosynthesis and takes place during vegetative growth. System 2, according to this system, ethylene enhances its production and takes place during climacteric fruit ripening and senescence of petals in some species. In Arabidopsis, upon various abiotic stresses, ethylene biosynthesis is enhanced via increased transcription of distinct subsets of ACS genes. Transcript level of ACS6 is increased in response to ozone. The expression of ACS2, ACS6, ACS7 and ACS9 are elevated during hypoxia. However, anaerobic conditions result in reduced expression of all the ACS genes. The transcript levels of distinct subsets of ACS genes increase after wounding and in response to osmotic stress, high temperature and drought conditions. In addition to ACS transcription, ACS protein turnover also plays an important role in regulation of ethylene biosynthesis. In Arabidopsis, ACS protein is divided into three main groups based on their C-terminal sequences. Type 1, proteins consist of an extended C-termini containing 3 conserved serine residues that are targets for phosphorylation by mitogen activated protein kinase 6, as well as a conserved serine residue that is a phosphorylation site for calcium dependent protein kinase. Type 2, proteins have shorter C-termini that are targets for only CDPK. Type 3, proteins consist of a very short C-terminal extension that lacks both the phosphorylation sites. ACS protein is rapidly degraded by 26S proteasome via a C-terminal dependent mechanism. The disruption of ETO1 resulted in increased stability of the ACS5 protein and increased ethylene biosynthesis. Binding of ETO1 catalyzes the addition of ubiquitin moieties on the ACS substrate. Then the substrate is ubiquitinated by the ligase and targeting the protein for degradation by the 26 proteasome. The stability of ACC is dependent on the phosphorylation of the protein. Phosphorylation of ACS proteins blocks the binding of ETO1 or EOL proteins to ACS, thus inhibiting the ubiquitination of these ACS proteins and their degradation by the 26S proteasome. Until now, we have seen that ethylene is controlling several aspects of plant growth and development. In addition, it is also interacting with other plant hormones, namely auxin, gibberellin, jasmonic acid and salicylic acid. Ethylene production is enhanced by exogenous application of auxins. Auxins upregulates the expression of ACC synthase 4 via modulating the expression of AXR1 and AXR2 and induces ethylene biosynthesis in Arabidopsis. However, in mung bean it enhances the expression of both ACC synthase and ACC oxidase. In Arabidopsis, transcription of 8 out of the 9 ACS gene is upregulated by auxin. On the other hand, ethylene regulates auxin level and auxin transport. Ethylene mediated upregulation of hookless 1 gene expression that facilitates apical hook formation is achieved by modulating the levels of transport of auxin. The complex interaction between ethylene and gibberellic acid demonstrated both negative and positive reciprocal effect. The mode of interaction between ethylene and jasmonic acid is determined by the developmental and environmental circumstances. Antagonistic interactions between ethylene and GA inhibits growth in Arabidopsis. The inhibitory effect of ethylene on plant growth and the interaction with GA is mediated by the DELA proteins. On the contrary, GA promotes ethylene responses, namely apical hook formation in the dark and hypocotyl elongation in the light grown seedlings. In oxygen deficient condition, GA positively influences ethylene activity. In submerged condition, reduced oxygen level induces ethylene production. Increased ethylene level inhibits abscisic acid synthesis. It is evident in Rumex that submergence induces the expression of GA3OX gene, which encodes the enzyme responsible for the production of active GA. Therefore, during submergence in one hand, ethylene reduces ABA biosynthesis and on the other hand enhances GA accumulation. The change in the balance between ABA and GA results in a GA induced stem elongation. Most of the studies showed that ethylene and JA signaling pathway interact synergistically. Both ethylene and JA signaling are required for the expression of defense related genes. After an attack by herbivore or a necrotrophic pathogen, both ethylene and jasmonic acid are increased. Elevated jasmonic acid rapidly conjugated with isoleucine and binds to carnitine insensitive 1 and facilitates COI1 dependent degradation of JAS repressor protein. This releases the repression exerted on IN3 and EIL1. IN3 and EIL1 from both jasmonic acid and ethylene pathways and activates JA and ET responsive defense genes such as plant defensin 1.2 or PDF 1.2. However, ethylene negatively regulates salicylic acid signaling. As mentioned earlier, after release of the repressor from IN3 and EIL1, they bind to the promoter of the salicylic acid biosynthesis gene SID2 and repress its expression and subsequently impairs salicylic acid biosynthesis and expression of pathogenesis related protein PR1. Now to summarize, ethylene is an important plant hormone. Chemically, it is an unsaturated hydrocarbon and synthesized in all plant parts. Methionine is the precursor for ethylene biosynthesis and the formation of ACS is the rate limiting step in ethylene biosynthesis. It plays a significant role in diverse cellular processes and also interact with other plant hormones like auxin, gibberellin, jasmonic acid and salicylic acid. Thank you.